I guess I'm introducing myself. So thanks everyone for coming to the Tools and Technology Seminar series. This is the last seminar before we take a break for the summer. I have already started scheduling some people for the fall, so if you're interested in giving a talk in the fall, please email me. It's always much easier when I get volunteers versus having to coerce people to talk. Since I do know there are some people online who are not usually at the seminar series, this is a seminar series talk about new tools, technologies, methodologies. They're either newly developed, recently developed, or simply of use for a lot of researchers. And so I'm Marci Brandenburg. I'm the bioinformationist here, and I will be talking about NCBI resources. I did get some backup today, so we do have some NCBI staff remotely logged in, so feel free to ask a lot of questions. If I can't answer them, hopefully they can. I'm also hoping they can maybe correct me if I say anything incorrect. So this seminar really came about because I recently took a couple of classes to learn more about the NCBI resources, and Brian asked if I would give one of these seminars to talk about what I learned. So as a result, I'm not an expert. I'm still sort of trying to process a lot of what I've learned, which is why several of the NCBI staff graciously offered to, or were willing to log on. So I'll start just by talking a little bit about the training classes that I took. And then I'm going to just sort of do more like a survey of resources, some of the resources that I've learned about. And I've just sort of broken them into categories here. I know some of these resources many of you will already be familiar with, so I'm not going to spend a long time on any one resource. And for those that I think you're probably more familiar with, I'm going to try to point out some things that you may be less familiar with those resources. And then towards the end, I'll just talk about what I hope to do in terms of library services in the future related to NCBI resources. So the two training courses, training courses that I took were sponsored by the National Library of Medicine, the National Center for Biotechnology Information, and the National Network of Libraries of Medicine NLM Training Center. These were specific for librarians, and they were free courses. For both of the courses that I took, you did have to apply. There were separate application processes and get accepted to attend the courses. So the first of the two courses was titled Fundamentals of Bioinformatics and Searching, which Scott nicely took with me. We took this in the fall of 2013, and it was a 4-week-long online course taught by Diane Rain from the University at Buffalo. She's the librarian there. The goal of this course was to provide an introduction to bioinformatics theory and practice in support of developing and implementing library-based bioinformatics products and services. This course covered basically molecular biology basics and an introduction to several of the NCBI databases and tools. During these classes, there was really, for people to attend, there was no expectation of having a biology background. So they did do a lot to try to get those people up to speed. And the format of this course were recorded lectures that we had to view, articles that we had to read, and then exercises that we had to do. This course, this online course, was a prerequisite for the Librarian's Guide to NCBI, which was a 5-day in-person course held at the NIH last month. And it was taught by NCBI staff and Diane Rain. The focus of this course was to sort of provide an overview of NCBI resources that are used to find molecular data. And the format for this course was 2 modules per day. Each had a lecture component and then sort of a hands-on component. And the hands-on component usually consisted of a demo that the instructor did with the attendees, walking through it together, and then some hands-on exercises that we were just sort of doing on our own with them available to answer questions. These are the topics that were covered in the Librarian's Guide. As you can see, we covered advanced entree, sequences, assemblies, and genomes, BLAST, sequence variation, gene expression, structure and function, drugs and small molecules, and then some additional topics. So I'll be briefly touching upon a number of the resources that we covered in these various categories. All right, so I'm going to start by talking about NCBI resources for genes and genomes. This, of course, will start with gene. I know many of you in this room are very familiar with gene, so I won't spend too much time on it. But gene, which was formerly known as Entree gene, covers, of course, gene-specific information. It has genes for many species. And it does contain unique identifiers that are used throughout many of the NCBI databases. You can, of course, get a lot of very useful information from a gene record. So you can get information about sequence, expression, structure, function, citation, homology data, and more. This is just an example of the top of a gene record. This is the record for estrogen receptor 1 for human. I basically just want to point out that on the right-hand side, there's a nice table of contents. This can give you a very quick idea of the type of information that you can get from a gene record, and will also link you to that particular area in each record. So if you know that you want a pathway information, you could just click on that, and it'll take you to that portion of the record. Just a couple things I wanted to point out from a gene record, of course. You have the location, the genomic context, so the location of the gene on the chromosome, and it will show you those genes that neighbor it too. And then I also just want to make sure you're aware there's a bibliography section in the gene record, so you can get articles in PubMed that are related to your particular gene of interest. So that's really all I'm going to talk about in terms of a gene record, because again, I know that most of you in this room are probably very familiar with the gene record.
Marci?
Yeah.
Could you back up for a second?
Yeah.
The bibliography articles, are those most recent first, or are those 5? Presumably those are not curated in terms of those 5 being shown, or?
I don't know the answer. Let's see if anyone online happens to know that. I mean, I know it'll link you to all of them, so I would assume that those are the most recent.
The publication dates.
Yeah, I know. So I was looking at that too.
Recent, so.
They're all 2013, 2014.
Okay.
Yeah, I'm not sure exactly how they do that. Peter's thinking. We'll see if he has got an answer to that.
He's going to let you know in a minute.
Excellent. So we can wait and see what the answer is to that. They do link you, of course, to all the citations in PubMed, but that's true. On the record, there's only a few available. I'm not sure quite how they do that.
I'll just read his response when it comes up. Don't
here we go.
Okay. Default PubMed order.
Okay. Here might be. Thank you, Peter. That's really all I'm going to talk about with the gene record. So what I want to focus on is something you may be less familiar with, and that is ways to search gene. And that would be field searching and index searching. Gene is actually a really great resource for doing field searching in. So some great field searches would be to search in the organism field. So I have an example here. I have yeast and then organism in brackets following that. So you'd put in the search box. So this is saying search for yeast specifically in the organism field. Another example would be there's an official gene symbol field. So you could do a field search for the official gene symbol. So I've got CFTR. And then you just put symbol in brackets following your search term. And that is saying search for this term, which is basically CFTR, specifically in the official gene symbol field. So really one of the best searches that you can do in gene, if you know the official gene symbol that you're looking for, and you're looking for it in a particular species, would be to do a combined search like I have down here. So this would be CFTR symbol, and is your Boolean operator. It does need to be capitalized. And then you would do your species, so human in the organism field. So basically what I'm saying is find all records that have CFTR specifically in the official gene symbol field, and it also has to have human in the organism field. And so if you do that correctly, you should get one result, and it should be exactly what you're looking for. In addition to field searching, yeah?
Where can you go to find out where all the possible fields are?
I believe that should be in the help documentation, so it's probably in the bookshelf information for gene. There's usually, like I know for PubMed, there's a whole area for field searching. But there's another way, and actually I'll be showing you that right now, because you can sort of get that information from index searching, too. So index searching is available under advanced option, which you can get, you can access that underneath the search box, and I'll show you that actually on the next slide. And just some examples of what you can do under index searching are you can search for organism, exon count, chromosome, etc. Like exon count would be, for example, if you wanted a gene that had a particular number of exons on it, you could actually search by that. So to show you a little bit about index searching, you would access it from this advanced option underneath the search box. And when you click on that, you get to your search builder. So here would be your fields, Maureen. So this would actually be the answer to your question. You have this all fields drop-down menu, and if you expand it, you get all the fields that are available in Gene. So I guess that would be the field information. That doesn't necessarily tell you what you put in the brackets, per se. So that, my guess is you would need to get that from the help documentation.
Is it build it in the URL or something? Like if you select one of these, can you
yeah,
infer
so
what
if
to
you
put in
build
it?
it'll actually, this prevents you from actually having to do that field searching with knowing the brackets. This is basically a way of
But
doing
it'll spit
field
out.
searching.
It'll spit out the bracketed version.
Correct, it'll recognize it for you, yeah. So you could choose like
Look at that.
organism here, put the organism in, here's your Boolean operator for and then you could choose the official gene symbol field, put in there, and it's the equivalent of doing that. You're searching specifically in those fields.
And then it'll build the manual expression in
Yeah.
the search box at the top, so you can see how it would have
Yeah, it
looked.
would put it up here, I guess. Does that make sense? So to do index searching, which is slightly different, you could choose a field, so for example, organism, and there's sort of two examples that I'll explain. One is, say you're searching on organism and you know the organism the name starts with HO, but you're not quite sure the rest of the species, how to spell it. You could put the HO in the search box, click Show Index List, and what it would do is it would give you, basically drop you right into the index of all the species in gene that start with HO. But what you may be even less familiar with is that you actually don't have to put anything in that search box to do index searching. So I could choose organism, leave the search box empty, and click Show Index List. And when I do that, I get the full index of species. So of course, it's much longer than this shows here, but it's alphabetical. It'll show you the number of parentheses as the number of records that are in gene for that organism or species. So this can be really useful. Say you want to know all the organisms that are indexed. Here, say you're going to do a number search, like exon count or something, and you're not really sure, well, how do you input a number? Do you write out the number? Do you put the digits? Do you need zeros before it? Do you need a decimal point and zeros afterwards? If that's the case, you're not really sure how to input it, you could go do index searching and you'd see how it's actually indexed within the database. So this is actually not only species, I take it back, it's all taxonomic categories. Thank you, Peter. So I guess that explains that. So that's index searching. I didn't know about it before the class, and I thought it was really useful and a good way to find out sort of how things are indexed and what information is actually in gene. So moving away from gene, I'll quickly talk about GenBank versus RefSeq. So both of these are annotated collections of publicly available DNA sequences and protein translations. And both have sequence records that could include a single gene or be a complete genome across different organisms. So how are they different? Well, GenBank is where the researcher goes to submit their sequence. The GenBank record is exactly the information that the researcher submitted, and it's annotated by the submitter. So say a submitter goes in and you submit some information and you type it in wrong, then the GenBank record is going to have it wrong. So as a result, GenBank records can have incomplete and incorrect information and records. They can also have redundant records. So you could have multiple people submitting the same sequence, in which case you could have multiple records for the same sequence. RefSeq, in contrast, is the reference standard. So these are records that are annotated by NCBI. They're updated by NCBI, so as things change over time, they can always be the most accurate and most up-to-date. And they're non-redundant. So whenever possible, you're going to want to use the RefSeq record and not the GenBank record. This is just the accession series for RefSeq, in case you're interested. If it's a genomic sequence, it'll start with NC, AC, or NG. If it's mRNA, it will start with NN, NR, XM, or XR. And if it's a protein, it'll start with NP and XP. And so each one does mean something different, and I could tell you that, but you can also just look that up. So if anyone's interested, I do have that on my little cheat notes here. So I could tell you, but they are differences, but these are the most important things right here. So all records that are in RefSeq also end up in either nucleotide or protein. So in most instances, you don't actually need to search RefSeq directly. You could just search in nucleotide or protein.
So remind me about the N and the X, for example, in the protein of the X. Is that predicted?
Yeah, yes, exactly. So NP is a protein associated with either the NM or the NC accession numbers. XP is a protein predictive model. And it's the same thing actually with some of these others. So like XM is a predicted model, as is XR, and one would be mRNA. So XM, that's mRNA, as is this one. The R is a non-coding RNA. And for genomic, the AC is a complete genomic model, but usually an alternate assembly. NC is also a complete genomic molecule, but it's usually a reference assembly. And NG is the incomplete genomic region. Any other questions about that? So I just want to take a moment to mention the International Nucleotide Sequence Database collaboration, because I think it's a pretty neat collaboration and something that is relatively rare. So if you are going to submit a sequence, you can submit your sequence to one of 3 databases. You can submit it to GenBank from NCBI. You could submit it to the European Nucleotide Archive through EBI, or you could submit it to the DNA Databank of Japan. Regardless of which of the 3 databases you submit it to, it becomes searchable from all 3. So they actually have a collaboration saying basically regardless, we're going to share basically records. So it doesn't matter which one you submit it to, it'll become searchable and findable in all 3. So I think that's sort of a neat collaboration and something you don't see very often.
Marci?
Yes.
Does that mean that they share nomenclature in terms of accession and other numbers, or are molecular entities reaccessioned to fit the native system when they come into, say, GenBank from EMBL and DDBJ?
That's a good question.
And Peter's
They have their own accession numbers.
Do they? Okay.
But they have cross-references to the other databases.
Yeah, so I'm guessing you can see which it was actually submitted to, would be my guess. I can't say that I specifically looked, but.
This is true for SRA as well? I think that's part of the GenBank collection.
Yeah, I assume so. Let's see. Yeah, so Peter confirmed that each database has its own sort of set. Might be getting another online thing. While we're waiting for that to come up, I can move on to genome-related databases, which will be the next thing I'm talking about that NCBI has. So I'm guessing that those are the information that you want. Thank you, everyone, online. It's a good thing I had backup help. So Peter's typing. While we're waiting for Peter to type, I can start moving on to the genome-related databases. And these are bioproject, genome, assembly, and biosample. So I'm just going to very quickly, it's the only slide I think I have on this, but try to sort of explain the relationship between these and sort of the differences. So bioproject is a collection of biological data that's related to a single initiative or project. So this could be all the sort of information that came from a specific collaboration or consortium or a particular organization. I'm going to stop real quickly because Peter wrote something. So the accession number is actually the same across all 3 of the databases based on that collaboration. And if you want the information about that, we have a nice link now that you guys could use to get that information. Thank you again. So bioproject is sort of collecting all the information that's sort of related because there's all from an organization or a collaboration or consortium. Within a bioproject, within a specific project, basically, maybe you have work that's being done in multiple species. So genome collects whole genome information by species or organism. So if that's the case, you're going to have separate records for those organisms, but they may all be attached to the same bioproject. Assembly collects information about structure of assembled genomes, and it will track the changes that take place to those genomes. And then biosample, I think, is more on the sample level. So it collects descriptions of the source materials to the experimental assays. So I borrowed this image from one of the slides from the course. As you can see, so bioproject is really sort of like the umbrella information. It has all the information related to the project. And then, underneath that, you sort of get genome and assembly and biosample that breaks it out into different organization in detail. I want to make sure you're aware that NCBI is a genome remapping service. So if you are working on a build of the genome and in the middle of your project, a new build becomes available. If you want to migrate over, you can do so using their remapping service. What this will do is it'll take the coordinates that you're using on the old build, and it'll remap them to the, or translate them, I guess, to the appropriate new coordinates on the new build.
So does this, I mean, I assume this is equivalent to liftover, but I mean, I assume that are they 100% the same, or are they 99% the same if I lift over a genome with one versus the other if I'm getting the same results?
I'm not familiar with
So liftover is the UC Santa Cruz genome browser, the way that they update the coordinates on their tracks and stuff. So I'm just curious how consistent that is, because a lot of people use that as well. And I mean, I assume they're using similar chain files.
I would assume they're fairly equivalent. If not, that would sort of concern me.
There'll be some regions that don't match. Okay, that's good to know.
Interesting. All right. Very good. Yeah, it would be interesting then to know actually what percentage is.
I mean, I'm sure it's pretty close. But
yeah, I would assume so, or else it would be difficult, I think, to justify using one over the other if they're not that similar. All right, so I'm going to move on to resources for sequence similarity, and this is, of course, BLAST. So BLAST, as I'm sure most of you know, stands for Basic Local Alignment Sequence Tool. It's actually BLAST2. A number of years ago, they redid the algorithm. They renamed it to BLAST2, but most people still call it BLAST. BLAST, of course, finds similar sequences to your query sequence. When you do a BLAST search, you could put the full sequence or a portion of the sequence into the search box, but you could also just use an accession number if you knew what that number was. I'll talk a little bit more about this, but you may want to increase the maximum number of target sequences that you're going to get from your BLAST results. The default is actually pretty small, and so you may be missing important information if you don't increase that. You can save in BLAST. You are saving the search strategy, though, not the results. So I want to make sure you're aware of that. And because, of course, things are always changing, if you run your BLAST search again, say 6 months from now, you may get very different results. So just know that when you save, it's just the strategy and not the results. And the last thing on this slide is the URL. I just got an email about this maybe a week ago. If you're using BLAST, make sure that you're using the first URL, blast.ncbi.nlm.nih.gov, and not using ncbi.nlm.nih.gov/blast. Starting in December, I believe, this latter URL will no longer work. So if that's the one you're using, you may want to change your bookmark or change which one you're in the habit of using. So, of course, researchers use BLAST to locate mutations and genetic variations, to compare 2 or more sequences, to find similar sequences from different organisms, to identify protein domains, to locate 3D structure records, and more. So this is just sort of your basic protein BLAST search window. So it's always a good idea to click the reset page button and also the clear button before you put in your sequence. Apparently there have been some stories where people have not done that and they've put in their sequence and they've run BLAST and they've gotten results that have nothing to do with their sequence. And they clear their cache and it still happened. I'm not really sure why that would be, but better safe than sorry. It's a good habit to just automatically click these even if you haven't run your search anytime recently. So when you run a protein BLAST, of course you take a sequence such as the one on the screen, you put it in your search box, or you could do the accession number. And then you have a few choices. You would choose which database you're going to run BLAST against. So the top one, which is the one that is currently selected, the non-redundant protein sequences, or NR, this is the most comprehensive of the databases. And it actually includes many of the things below it. So it includes SwissPro and Protein Data Bank and GenBank and stuff. There are some algorithms that you can choose from. I'm not really going to go into this other than to say that BLAST-P, which is the protein-protein BLAST, that's the default. I think that's probably the most basic and most common algorithm to use. If you want more information about the other algorithms, there's this nice little question mark down here. You can click on that and it'll explain the differences between the algorithms. Down at the bottom, to the right of the BLAST button, there's an option to check a box so that your results will show up in a new window. This is always advisable so that you don't lose your search screen and you don't accidentally click out of your results, something like that, and then lose everything. And before you click the BLAST button, you may want to look at the algorithm parameters, which are at the bottom of the screen. And if you expand that, you get this window. And there are a couple of things I want to point out here. The first is that maximum target sequences. So this is what I was talking about earlier. You may want to increase this. The default is 100. You may want to increase that to 1,000. I think you may even be able to increase it to 10,000. I'm not sure. But 100 is sort of low. And I'm going to show you in a minute ways to filter from your results so that you can narrow down your search. So you may as well pull in more results initially and then filter from there to make sure you're not missing anything important. The other thing I want to point out is the short queries option. So the BLAST algorithm is meant to work on queries of or sequences of a certain minimum length, but you can actually run BLAST on shorter queries. But if you do so, you just need to make sure that this box is checked so that it will rework its algorithm appropriately. So once you run your BLAST search, you get some results. One of the things you get is this descriptions table. I just want to mention the e-value if you're unclear about what that is. E-value is the probability that your hit or result is random. The best score is 0, basically means it's not random. A value greater than 1 means it may be random, and a value greater than 10 means it's likely random. The column to the left of e-values, this query discovery, that is the percent of the sequence that is exactly the same. You also get some conserved domain information. So the points of these mountains are where the conserved domains are located. Although it's not in this example, sometimes in this query sequence information, you'll get some areas that have sort of a turquoise color. If you get that, that represents an area of low complexity. And you get your distribution of your BLAST hits. This defaults to doing about 100 hits. There's a way in the filtering options to actually increase this. So keep in mind, these are not necessarily all your results. It's just doing a small number of them. So if you increase your results, say like 1,000 hits, this might only show a portion of those because it's just quicker for the computer to compute. You can see the color. There's a color bar here. So all these are pretty well aligned, but sometimes you'll see portions of the bars or full bars of different colors. And if this were live, I could take my mouse and scroll over each of these bars, and the information up here would change to represent the bar that I'm currently on in that sequence. So I want to talk about formatting options. On your BLAST results page at the top, you have some options, one of which is formatting options. And if you click on that, it'll give you this information. This is a way basically to filter your result set to get more specific results or information. So one thing, which I'll talk about in the next slide, is you can change the alignment view. But you can also do some things, again, to sort of filter. Say you want only results of a particular line length or only a particular organism. You could filter that here instead of doing it early on. Or you could exclude an organism. Say you only want results that had a specific percentage identity match with your query sequence. Well, you could do a minimum and maximum here. So this is really great, and apparently a lot of people are not familiar with this and don't know this is here. So once you have your long list of results, this can help you narrow down to exactly what you want. So I want to talk about the alignment view. So the default view is pairwise, which looks like this. If we look at these first 3 rows at the top, the first row is our query sequence. The third row is our subject, or our result, basically, sequence. The middle row is telling us whether or not they align. I find this very hard to read. I feel like you have to look at it really closely to see where it's aligned and where it's not. But you can change this view. So one example, there's several different views you could change it to. But one is called pairwise with dots for identities. So here, this top line, again, is my query sequence. The bottom line is my subject or result. If it's got a dot in the result, that means it's aligned with your query. If it's not aligned, it shows you the difference, and it's in red. So I find this much easier to read. So I want you to make sure you're aware there are different ways that you could change this, so you don't have to necessarily look at this, in my opinion, more difficult view to see the alignment. All right, so the last thing I want to say about BLAST is if you use BLAST, you do need to cite it. So you should be citing this 1990 article if you use BLAST for your work.
How many citations does that have now?
Probably 8 gazillion. Well, we hope 8 gazillion because we hope that people are using BLAST, they're citing it. All right, so I'm going to move on to gene expression, which of course is GEO. GEO is the Gene Expression Omnibus, and it is the data repository that has array and sequence-based data. So GEO has GEO dataset, and it has GEO profiles. So GEO dataset is curated, and it includes what's called a series entry, a samples entry, and a platform entry. And I'll talk more about those on the next slide. I want to make sure that you are aware that GEO dataset only has array data. There's no RNA-seq data in GEO dataset. And GEO profiles, that consists of the expression measurements for an individual gene across all of the samples in a GEO dataset. So GEO is Miami compliant. That is a requirement of many journals. So if you have that requirement, you can submit to GEO, and it will meet that need. When you submit to GEO, you're going to submit general information about what the study is. So what you did, why you did it, how you did it. All that information goes into what is called a series record. You will also submit sample specifics and the data that you obtained. That information goes into what is called a sample record. And then you will also submit information about the array, and that goes into what's called a platform record. And all of these can make up a GEO dataset record. There are more series records than there are dataset records, because when you submit information, it's a series record. Remember, the GEO dataset records are curated, and so curation, of course, always falls a little behind since it takes time to do that. So here's an example. I searched for GSE 4523 in GEO datasets. Even though I searched basically one accession number, I got 9 results. And you might be wondering, well, why did you get 9 results when you did such a specific search? Well, the breakdown's on the left-hand side. One of these is a dataset record, one's a series record, 6 are sample records, and 1 are platform record, all related to that same accession is what I'll call it. This top result, I believe, is the dataset record, and you can see right here in the information, it gives you the information for the platform, the series record, and it tells you right there are 6 samples. And it links out to GEO profiles too, so if you need the individual expression information, you can access it there. All right, so we move on to protein structure. Yes,
You
Maureen.
go back.
Yes.
So GEO used to have this really nice query page where you could query like this. There were different query boxes. I don't know. And I can't find that page anymore. I always get to this one.
Yes.
Can you get to that other page still? Or
I
is
don't
that
know, but
completely gone?
I
Is
haven't been
anybody
able to get to
online?
that page either, so maybe someone from NCBI
You know what
can
I'm
help let
talking
us
about.
know if that will ever be coming
missed it.
back. Or, but yeah, I found the same thing, because every time I did a search, it basically dropped me into GEO data sets.
It helps you. You can probably do everything here that you could do there, except you have to know what you're doing more here. It's more user friendly.
Yeah, and I know there's a browser link from the GEO homepage, but that also is just browsing data sets as far as I know. So I have experienced the same thing. I don't know if that removal of the other one is a permanent removal or not. While I'm talking, we can see if anybody decides to respond. We may not have any GEO experts online. Let's see, we have typing at the moment. It's a very good question. That is one thing about NCBI. Things are constantly changing. Trying for the better, but
Yeah, it looks like they've tried to integrate GEO into NCBI and other stuff.
So are you referring to the former limits page? Here?
We're probably talking about the same thing.
Yeah.
It had, like, if I Googled GEO, that used to be the main page that would come up. And then there would be, like, 3 different boxes. Like, you could either search for a series, search for platform, search for,
Yeah, it's gone.
Okay.
But if
For a
that is
while,
something
both
that
of
you
them
really
were up there.
missed, that is good for them to know.
Yeah.
Okay, so you find the new page, it sounds like, more complicated than the old one. Or less user-friendly.
Yeah.
Okay. That's always a good
Well,
feedback.
they're both available.
Then maybe that was during a transition period, yeah. While Peter is typing, I will just move on. Oh, no. So yeah, so it looks like that's the replacement page, yeah. All right, so move on to protein structure. So structures do not get submitted to NCBI. They get submitted to the protein data bank, and then they become searchable, or their records end up in structure and protein and other resources. So down at the bottom, I have the top of a record in structure. I do want to point out that on the right-hand side, they do have information here for when it was deposited into PDB, the experimental method, and some other just useful information right there at the top. If you scroll further down in the record, you will get to a box that looks like this. And this is how you would view the structure in 3D. So NCBI has 3D viewing software called CN3D, which I'll talk about in the next few slides. This is a software that needs to be downloaded and installed on your computer. So if you don't already have it, you could use this button right here to download it. If you do already have it on your computer, then if you click View Structure, it'll launch the software on your computer with the structure for the particular structure in that record that you want to view. So exactly what is CN3D? It is, again, NCBI software to view 3D structures. It works with Windows and a Mac, and it can be compiled in Unix. It shows you the structure, sequence information, and alignment information. You can create annotations, and if I remember correctly, I think you can actually view them on the image, and you can change visual properties, although I won't be showing you that today, since I'm just going to go through everything really quickly. And you can also export the image as a PNG file. So this is a structure in CN3D. Of course, if this was live, I could use my mouse and change the orientation of this and move it around and zoom in and out. I'm guessing, Barry, is this sort of standard stuff that like the cylinders represent alpha helices?
It is.
Yeah. So I figured that was standard, but the cylinders aren't real. They represent alpha helices. The spaghetti stuff is backbone.
The spaghetti looks messed up though.
Interesting. So you get your image up here. You get your sequence and alignment viewer down here. This screenshot was taken on a PC. So in a PC, you have separate menu bars for these two windows. Apparently in the Mac, it's one menu bar, so you have to be very careful about which one you're actually clicked on at that time. There are different views you can view. So the worms is default. It's actually the least accurate, but it's the easiest for the computer to draw, so it is the default. But you can change it by going in the Style menu, rendering shortcuts, and then you can choose from these options. So this is actually space fill, which you can see looks very different from the worms, which was what was here. And the last thing I'll mention is that you can select a portion of the sequence in the sequence viewer. So here, what's in yellow, I selected that, and it will map that and select it in the actual 3D image. All right, so I'll move on to small molecules, unless there are any questions about that. All right, so this is, of course, PubChem. There's PubChem substance, PubChem bioassay, and PubChem compound. I always found this extremely confusing and never understood the difference until I thankfully attended this class. So PubChem substance has substances of known or unknown composition. It has real chemicals. It also has some very odd things like chamomile tea, where someone hydrolyzed a feather and then submitted it to substance. PubChem bioassay has experimental information that's related to the chemicals in substance. Now, in contrast, PubChem compound is information that comes from substance, but is curated and annotated by NCBI. And they've also added a number of valuable internal and external links to those records. So again, when possible, you want to use compound. It's also going to be the most up-to-date, so if a compound, a chemical changes name or something changes in it, it'll be updated here. So just to make sort of an analogy, I sort of see PubChem compound as being the PubChem substance as GenBank, as RefSeq is to GenBank. So when you submit to PubChem, you must sign an agreement saying that anyone can freely use your data. So you should know that. PubChem is continuously updated. So if you submit a chemical and you would say make this available as soon as possible. It will usually, you should see it within 1 to 2 days. So this is just the top of a PubChem record. PubChem records, compound records, can get very long. There's a table of contents on the left-hand side. This, of course, is only a portion of it. But again, it can link you to particular parts of the record that you're interested in. PubChem does have a limits option. So if you use PubMed, you may have noticed if you used to use the limits option, it's no longer there. Now there's a refined search option down the left-hand side. Well, in PubChem, limits does still exist, and it is going to continue to exist as far as I know for the foreseeable future. So some things that you can do with the limits, you could limit to chemical properties, such as chemicals that have particular molecular weight, particular stereochemistry. You can even limit to particular elements if you want to make sure they're included. PubChem is supposedly actually one of the best documented resources in NCBI. So if you have questions, I recommend looking at their documentation. The last thing I want to mention in this slide is these sort of icons up at the top right. So some of those are for downloading information, but the one I want to point out is the structure search tool, which is second from the left. And if you click on that, it'll take you to the chemical structure similarity tool. And this is where you can go to search on a structure for similar structures. So for example, I have here a chemical ID. I click the edit button, and it takes me to the sketcher window. So at this point, I could edit the structure and then search on the new structure that I created. Or you could actually get a blank canvas here and draw your structure. So say you had an unknown structure and you didn't know what it was and you wanted to find out, you can maybe draw the structure here using these nice tools over to the left, and then search against the known structures and come up with, try to figure out what it is. All right, so I'm going to move on to additional resources. These are just a few additional things that I thought are worth telling you about. So one is SciENCV. This is pretty cool. So if you have an ERA Commons account, and those of you in this room know who you are because that's the system that you use to manage all your grants, you can link that to my NCBI, which I'm guessing all of you have. SciENCV is in my NCBI. This stands for Science Experts Network Curriculum Vitae. And their hope is to eventually make this a federally wide biosketch repository so that you can have all your information in one place, and then regardless of which federal agency you are submitting your grant to, you'd be able to get your biosketch in the right format using this system. Right now, however, the only output is your NIH biosketch. This is very new. This just came out a week or two ago. You can now create multiple profiles. So say you had multiple NIH grants and the NIH biosketches were slightly different, you could save them as multiple profiles. The input for this is ORCID. If you have that, if you know about it, it's a unique identifier to help disambiguate authors. So if you have an ORCID ID, you could use that as some of the input. You can input information in your ERA Commons account and also in your My Bibliography information. All of that goes in. You can also manually add information. And then the output is the NIH biosketch. So this is what it looks like. I went into my NCBI to the SciENCV. And so you can see right now I haven't put any information in this example, but you can see the normal breakdowns, your education training information, your personal statement, positions and honors. And then at the top right, you can download in the correct format as a PDF, Word, or XML. This is a very recent change. Up to just about a week or two ago, it was only a PDF. But this is very useful. So I went in after doing this. I added some of my information just to see what it would look like. I downloaded a PDF, and this is what I got. In perfect NIH biosketch formatting. As someone who worked on this stuff for the BD2K grant, I highly recommend doing this. You will hopefully make the lives of your administrative people so much easier.
You mean everybody doesn't follow the same format?
You know, what am I thinking? Of course everyone follows the same format. So I actually thought this was really cool and would have been really helpful for me to have known about when I submitted mine for the BD2K. So
the ability to save it as a Word document is excellent.
Yeah,
It's
so
something
that's-
that MCV here is really lacking. Because
yeah.
administrators like to copy and paste all
Well, that's what I say. I mean, Helen, I'm sure, will want it in Word. So they just add that in, and I think that's going to be key.
Makes it very useful.
Yeah. So I thought this was really cool. I actually didn't know about this before the class, and so I want to make sure everyone was aware of that. I also want to make sure you're aware that NCBI is a submission portal, so this is a really good URL to just keep in mind. It's submit.ncbi.nlm.nih.gov. So this will sort of link you basically to all the various submission locations. So you can see GenBank, Geo, NIMS, that's where you go to submit your NIH manuscripts to be compliant with the public access policy. So this is sort of like one-stop shopping here if you do a lot of submissions for NIH stuff. They have a new entree search. This came out since I returned from the class. This is also known as gQuery, and now it just says search NCBI databases. Basically, this is just how you do the federated search so that you would be searching in one place. So here I've searched for CFTR, and it searched it across all of the NCBI databases and will tell me how many results I would get in each one. I think this new interface is much cleaner, and I like it much better than the old one, so I want to make sure you're aware that that had changed, in case you were staying away from it because you didn't like the old interface. And the last thing I'm going to talk about is PubMed Commons, only because if you've been going into PubMed, you may have seen this and may be like, what on earth is this? So this is your normal PubMed homepage now, and on the right-hand side here, you'll see that it now says PubMed Commons, so you might be wondering what that is. So PubMed Commons is basically a commenting system. It allows authors to share opinions and comments on scientific publications that are in PubMed. You do have to use your name if you leave a comment, so that's one way that they've tried to use to try to help ensure that people are going to write legitimate information in there and comments. Any author that has a publication in PubMed can leave a comment. There is, at this time, no monitoring because NCBI does not have the staff for that, but there is a report abuse link, so if you do find something on one of your articles that is inappropriate, you can click that and you can get it removed.
How do they, you say any author who has a publication, so how is that linked, and how does that affect if you have multiple author names on different publications? And is this incorporating like ORCID ID or anything like that to resolve that type of thing?
I don't think it is.
I'm Mills RE on some papers, and I'm Mills R on a couple others, and I mean, I guess.
Well, so you do also create an account.
Okay, so you tell them which one to use?
Yeah, I think so. So if I go to this screenshot on the next page, you have to join PubMed Commons, so I'm guessing when you do that, maybe you are, I haven't tried it actually, so I don't know. I guess I should have tried that before coming here.
This was a beta test for a while, I think, because I know I tried to do it before. It was like select access, I think, at the time. So
Okay. I know that apparently they've been surprised by the number of people I think that have actually been doing it. If you do have problem, anyone, or you're not an author and you're interested, I may be able to help you get access to that. But yeah, so I don't know the exact answer, but I'm guessing that when you go through this process, you're probably identifying yourself and maybe your papers.
And so, and my other question, I guess, is, are any of these systems incorporating ORCID IDs? Because I know there's a push here in Michigan to get more people.
So I think ORCID ID may be in PubMed now. It's certainly in SciENCV. You can use your input and link it up there. I actually did that with mine. I went and logged into ORCID ID. So more and more of these systems certainly are. And I know they are trying to tie more stuff to ORCID ID. It seems to be picking up a lot of steam right now. So I think it's a really good resource, and I think you can use it. I know that, well, I'm pretty sure Scopus has it in there because that's Elsevier. So I know more and more of the databases are starting to incorporate it. Let me go back to the slide real quickly. So as of right now, authors are not notified when a comment is left on an article. That is a feature that they would like to add in the future, but as of right now, you would just have to go check your articles if you want to see if anyone's commented. There are guidelines if you're going to use PubMed Commons, and the one that I just want to point out right now is that comments can be freely used by anyone. So keep that in mind if you're going to make comments. So this is, yeah.
You need to join in order to see if any of your...
No. So this is a standard PubMed record now. I just went to PubMed, found this article. Here's your normal information, your abstract. Below that, now it says PubMed Commons. And you can see the comment right there. And there's something like Amazon.
So then can you search for most commented instead of?
I don't think you can. I
Popularity or
don't think
something?
the comments are searchable. That's actually a really good question. I don't think they are, at this time at least. there was a comment made in the chat. Right, so you have to be an author to comment. Yeah, so basically the PubMed Commons, those comments that are made are publicly viewable to anybody. You just go to PubMed. So you could look up your article, go to the record, and you'll see if there are any comments there. I know, because I check my articles. So lastly, I'll talk about library services, which is really just a slide. So now that I took these courses and I'm learning a little bit more about the NCBI resources, I do hope to teach on some of them. And I would certainly be interested to know which of these you think would be of most value for me to offer training sessions on. I also, of course, hope to offer some consultations, so one-on-one things or small group things. If someone has some data or is not sure which resource they should use, something like that, to sit down with them and talk that through, and also, of course, just answer basic questions. I have a nice in now at the NCBI group, and there's a nice listserv, too, actually, that with graduates from the course and the NCBI staff. So if I can't answer a question, I can submit it there and might be able to get help from NCBI or other librarians who may have more experience. I'm going to come back to this with resources. It would be of most value to teach, but I'll just finish through my slides. This is just a photo of all of us who attended the course and the instructors in front of the National Library of Medicine. And I would like to acknowledge and thank all of the instructors, particularly those who are currently online right now. And with that, I'd like to see if there are any more questions. Say thank you. And I would also like to come back, like I said, if anyone has any suggestions, just from your experience and knowledge of what resources you think would be most valuable to have training on here, I would love to hear that. Lisa. Oh, you can search comments in PubMed. Thank you, Marisa.
So that's the field?
Yeah, so that would be the field. As user.
Looks like that searches for presence of comments and not in the text of comments. But that's a useful thing to know. Search with your author name and then has user comments and find whatever articles of yours have been commented. Yeah, comments are not directly searchable this time.
Yeah, so you're not actually searching the comment, you're just searching whether or not the article has a comment.
Yeah.
But that's interesting. That's good to know if it's your own article.
Yeah, so if I was an author, I would save a search in PubMed that was my name or my ORCID ID or whatever with that field, and then if you can order them by last commented on, that would be a cool search to have, and then pull out occasionally to see what you've got.
And ORCID ID is a field now in PubMed, is that right? Is it a field? You guys know?
User submitted author identifiers are
identifier.
Okay, so that's what the comment is. Okay. All right, any questions or anyone like to provide input on training, future training?
Great, have your reviews up there.
All right, I guess not. Well, thank you, everyone. And thank you, everyone online.
Thank you, Marci.
Get access to all the reviews available.
Marci,
Yes.
send Maureen the field data.
I don't have it.
Thank you.
It's in the chat.
I figured.
And one.
Bioinformatics for me.
Well, the one that I did just
I gotta go back. Nope, I don't know if we're
I'm trying to see what the ORCID ID looks like. There's a PubChem idea. Because that could be useful.
I just downloaded the whole thing.
Now you have one.
Thank you.